We're going to continue learning about plants, and today we're going to introduce angiosperms. I have a beautiful angiosperm right here in my hand, right? Look at this. So these guys are going to have petals here shown in red. They're going to have sepals shown here in green. So the sepals, the petals, and they are going to have these stamens producing the pollen. It might be hard for you to see, maybe there, in my the background and my hair. They have these yellow dots. That's the pollen grain, or the pollen grains, and they also gonna have the pistil. And part of the pistil, we have it right here at the top, and it goes all the way to the bottom. At the base, we're gonna have some ovules. So I'm gonna destroy it for you. Maybe we'll be able to see. The ovary is right here. And I have some ants as well. But we have this structure. This is the ovary. And the ovary is gonna have ovules inside. So they are gonna be very small. I'm not gonna bother to open it further. But that's a beautiful angiosperm. So angiosperms are gonna be flowering plants. This is an example of another flowering plant. This is basil. It smells really good. So basil is used in Italian cuisine mainly, and it gives a really nice flavor to some of the dishes. They have also aromatic compounds, and you can see the tiny flowers compared to this hibiscus plant that I just showed you, the big red flowers. These are very tiny. Some other flowering plants might even have smaller flowers, very, very tiny. Today we're gonna learn what's an ovule and what's pollen or pollen grains. We're gonna identify the two main lineages or clades that form angiosperms. We're gonna distinguish the megasporangium and the microsporangium and understand the differences between gymnosperms and angiosperms. Also very important, we're gonna learn at least three main synapomorphies that distinguish angiosperms. What's a flower? We're gonna discuss a little bit about that, and we're gonna identify the structures of a flower. So I just briefly did with the hibiscus plant, but we're gonna cover it a lot more. Remember that we are covering land plants and a group that is called seed plants that is also a type of a vascular plant. And in this group, we're covering the seed plants that have flowers. And most of them are very beautiful. So seed plants, remember, are gonna be plants that have seeds, which is this structure that has baby embryo with food supply surrounded by a protective cover. And that protects it against biotic or abiotic factors. The other thing that I want you to remember is that seed plants have heterospory, or they are heterosporous. And I want to ask this question for you to see if you are understanding these concepts. Leaves that bear megasporangia form megaspore. And leaves that bear megasporangia are called what? A, megaphylls, B, microphylls, C, megasporophylls, D, microsporophylls, and E, gigaphylla. So remember that megasporangia are going to be there in these megasporophylls, right? Megasporophylls is the right answer, C. So do not get confused. Microphylls and megaphylls refer to the vascular tissue in leaves. So microphylls have one unbranched vascular tissue in these leaves, whereas the megaphylls are going to have branched vascular tissue. So that's the difference. In contrast, the sporophylls are going to be the leaves carrying sporangia. And we say that the megasporophylls carried megasporangia, and the microsporophylls carry microsporangia, or female and male sporangia. That's the difference. Okay, but what does angiosperm mean? What does angiosperm mean? So for that, I would like to define what does gymnosperms mean, and that comes from the Greek. So a lot of the words that we use in science comes from either the Greek or the Latin. And in this case, gymno means naked, and sperms actually means seeds. So gymnosperms have naked seeds. And that is because the seeds and the ovules, when they get fertilized, the pollen can reach the ovule directly, right? Right here, you see the pollen landing in this ovule. I say that some of them form pollination drops. Some of these gymnosperms can form a pollination drop. But then the pollen reaches directly the ovule. In contrast, in angiosperm, there is no such thing because the ovules are enclosed in this structure called the carpel or the pistil. And we'll see that some pistils have multiple carpels. And a carpel is just a modified leaf that is going to be carrying these ovules. So in angiosperms, again from the Greek angion, that means vessel, and sperms, meaning the seeds, that is that the ovules or seeds are going to be enclosed in this carpel, in this vessel. So you see, the difference is then that in gymnosperms we'll have the pollen reaching the ovule directly, whereas in angiosperms we have this stigma in which the ovule is going to land. Then in this area, it's going to rehydrate because the pollen gets really dry in order to survive. And then it rehydrates first and starts germinating. So it creates a pollen cube carrying the spermatic cells, the gametes, that they are going to reach at a certain point the ovules. So the pollen needs to grow all of this in order to reach the pollen, in order to reach the ovule. So that's the difference. So gymnosperms, naked seeds, and angiosperms are cover seeds, right? They are going to be containing this vessel. Angiosperms evolved most recently in all of the lineages that we have discussed, the large lineages. So if we look at this table with the geological periods, we can find the angiosperms evolving during the Cretaceous. And the Archaeofructus fossil that I have showed to you in previous classes, it has been dated, I think, 125 million years ago. So that's at least flowering plants occurred then, but the origin of flowering plants can be dated earlier. Flowering plants have more than 300,000 species, and as I mentioned, they can be, the lineage, it's at least 140 million years old. And this is one of the families that has a tremendous importance to humans. It's economically important. And for example, think about food sources. Think about some of the paper that you use. A lot of the cattle eat these plants, angiosperms. And some of the synapomorphies, or the features that distinguish angiosperms, features that are shared among all of the angiosperms and not other groups, are flowers. So angiosperms have flowers. They are flowering plants, right? But they also have fruits as a consequence. And the ovules, and therefore the seeds, because the seeds are the result of fertilization. So the ovule becomes a seed. These are enclosed in a carpel. And they are going to have double fertilization with triple endosperm. I know this is like, what's going on? We're going to cover that in a second. But then we're going to have also a specialized vascular system. So we say that vascular plants have vascular systems, and they are going to have xylem and phloem. But in angiosperms, the xylem is going to have vessel elements and fibers for example, in other plants, in other groups, we only have fibers, not vessel elements. And the phloem is going to contain companion cells. So in here, we have one of the cells in the phloem, which are called sieve tube cells. And the companion cells are these cells on the side that provide nutrition to this vessel, to the phloem. Flowers are going to be a specialized structure, the sexual organ in these angiosperms. And they can have multiple type of structures, up to 4 in this flower. But there are many differences in different groups. So when they have the 4 structures, they're going to have sepals. In this Arabidopsis thaliana, we have the sepals in the outside, in green, and later we'll have the sepals in white, and then the stamens, and then the carpel in the center of the flower. The stamen is going to have, is going to produce pollen, and the carpels are going to produce ovules. Sepals are usually green, but not always, and petals are usually colorful, but not always. So this is a different flower for you to realize the presence of the ovary, right? And in the ovary, we're going to have the ovules inside, because the ovules are protected. So this picture is taken from a plant that has been cut, so you can see part of the ovules. And the stamens are surrounding this carpel, okay? And then we have some of these sepals in this case. So, and look at these 2 flowers, for example. This flower has a superior ovary, right? The attachment of the stamens happens below the ovary, whereas in this case, the ovary is inferior, with the stamens that you cannot really see are attaching above the ovary, and we see these ovules. The stamens are going to have 2 main structures. One is the filament, and that's the stalk, and the other one is the anther. And the anther is the actual structure that is producing the pollen grains. In contrast, the carpel is going to have 3 structures in most of the angiosperms. We're going to have a defined structure called the stigma, and this is the area in which the pollen gets deposited and usually dispersed by, let's say, insects, although there are some angiosperms that get also pollinated by wind. But then regardless of the vector, the pollen will land in this stigma, and in this area, there are specialized cells that hydrate this pollen, and they also have some molecules that allow the pollen to grow. If the pollen is not recognized as the same species, or if they might be too similar, there are some species of plants that detect if the pollen is from the same plant and they don't like to have selfing, they don't let this pollen grow. So there are some molecular mechanisms that prevent selfing or crossing with other distant related species. Then the stigma is at the top, usually, and we have this style, which is this stalk that the pollen grain, and you can see the tiny pollen grain, and these few cells are going to elongate long to reach the ovule. This is a cartoon of the flower, similar to the one that I showed you earlier, right? This one. And, well, not that similar, but you have the idea. We have a structure called the receptacle, that is where all of these whorls will converge. And then we have the sepals, the petals in orange, and then the stamens with the filaments indicated, and the anthers. And please take a look to this carpel that has the stigma, style, and ovary. And inside of this ovary, we have the ovules. The ovule contains the megasporangium, which is diploid, and we have a megasporocyte. This megasporocyte is gonna go through meiosis to produce 4 megaspores, right? So megaspores are female spores. And we have 4, right? Result of meiosis, we're gonna have always 4 cells, but look at what happens. 3 of them die and only 1 survives. The megaspore is not released. So do you remember how in ferns, the spores get released to the air, and then they go and land in another area and produce a new individual? Well, or a gametophyte. In this case, the megaspores get retained, and they go through mitosis in the sporophyte, right? Inside of the sporophyte. So we have now, we have 1 megaspore here, and now through mitosis, we have formed this female gametophyte that has 7 cells and 8 nuclei. This is gonna be important later on. So remember, karyokinesis without cytocinesis can form 1 cell that has 2 nuclei. And this division, so we have 3 consecutive meiosis, and then 1 of these cells is gonna form the egg, right? This is the egg. And this is how it looks when we do cross sections of this megagametophyte. So here we have the egg. These 3 antipodal cells, don't worry about it, they disappear. Synergids seem to have a function to produce some molecules that will direct the pollen tube to reach this area. Let's see what happens in the male part now. So if we do a cross section of this anther, we'll see that inside we have a cell that is diploid, a microsporocyte. This microsporocyte is gonna go under meiosis and form 4 microspores. And these microspores they don't degenerate. In contrast to what happens in the female part, all of them get formed and developed into microspores that later on go through mitosis. You see how we have now 2 cells, and this is the pollen grain, or male gametophyte. Pollen grain is the common name of the male gametophyte, if you want to see it that way. This is the sporogenesis, the formation of the spores, right? They go from diploid to haploid is what we define as sporogenesis. And then the consecutive divisions, mitotic divisions, to form these gametes. The spermatic cell is 1 gamete, and the egg, it's another gamete. It's the female gamete. That's the gametogenesis. In gymnosperms, this life cycle, it's going to take, at least this part that I explained, can take 1 year or 2 years. If you remember in the previous class, I say that some conifers can even take 4 years. And in angiosperms, we complete the cycles very fast. In some of them, we have annual herbs that they germinate from the seeds. They form the flowers really fast, and the fruits can be ready in few weeks. And they just close the cycle very, very quickly. That's a selective advantage that the angiosperms have developed. And because of that, seeds produce rapidly, and fertilization occurs very soon after pollination, not like in gymnosperms that the fertilization can happen 1 year after the pollen lands on the ovule. And if you think about this, it's almost like if you have sex and you care, if you're a female, if you were carrying a sperm for a year and the next year you get pregnant. That would be the equivalent to this process. Analogous idea, and silly as well. Okay, now the sporophyte in angiosperms usually have both the structures, the male and the female structures, in the same plant. Most of angiosperms have the stamens and the carpel or pistil in the same flower, actually. They are called hermaphrodites or bisexual. the structure, the flower structure. Most flowering plants have a mechanism to ensure cross-pollination. We can think about pollination as self and cross. Self-pollination will be plants that allow the pollen from the same plant to fertilize their ovules. And this is one strategy that will allow one single plant to colonize an island, for example. And you don't need to mate with another plant, but you have the ability to reproduce with yourself, and well, in this case, the plant. And therefore, that's very successful for colonizing new areas. However, that can have some genetic consequences. And in those cases, cross-pollination has evolved. And some plants have strategies that, for example, they produce first the pollen or the anthers. And then once that the pollen is released, the ovary matures and they get receptive. So once that they don't have pollen of their own, they can receive pollen to fertilize their ovules. And there are other mechanisms. There are several mechanisms to ensure cross-pollination. But then, basically cross-pollination is the transfer of pollen grains from one plant to a different plant. One of the features that angiosperms have is double fertilization. And double fertilization is going to be due to the fertilization of the ovule. So as in conifers and gymnosperms in general, we're going to have the fertilization of the egg, and that is going to produce the embryo. However, in angiosperms, we have another fertilization happening in the life cycle. That's what we're going to learn now. This is the pollen grain which landed in the stigma. Remember, the stigma has these really nice juicy cells that hydrate the pollen and recognize the pollen. Once that they recognize it, we'll let it grow. Right? This pollen has the right keys to enter the house, and this pollen has haploid cells inside. So in here you can see two spermatic cells, or two sperms. They are not flagellated sperms, right? Then these sperms are carried by this pollen tube that grows and grows and grows, and it's going to reach this ovule. In the ovule, we have the integuments. Remember, the integument is this structure that is going to differentiate later into the seed coat, and that will confer protection in the seed. So this ovule is just an unfertilized seed, if you want to say it that way. It has the integuments, and it has this micropyle, which is a space in between the tips of the integuments, and this is the area by which this pollen tube is gonna deposit these spermatic cells. So you see how the pollen tube grows and grows and reaches now the ovule. It's gonna deposit one of these spermatic cells. It's gonna fertilize the egg. What happens to the other one? The other one comes to these polar nuclei. The polar nuclei, we have two nuclei there, and it's these nuclei are contained in the central cell. So this is not marked here, but this large cell is called the central cell. It has two nuclei. Then if we fuse one spermatic cell, which was haploid, and these two nuclei, which are haploid as well, what will be the diploidy of this cell? Haploid, B diploid, C triploid, D tetraploid. Okay, it's super simple because you can see it here. 3N means triploid. And this is the nutritive tissue, the food supply in angiosperms. If you remember, in gymnosperms, the food supply was part of the gametophyte. We say gametophytes are always haploid. So the source of nutrition for the embryo, the first food that the embryo is going to taste when that the metabolism resume, is the haploid tissue. In angiosperms, this is coming from the endosperm, which is triploid. These are the only group of plants that have this in their life cycle, this double fertilization. Let's talk about this endosperm, this triploid endosperm. This is the nutritive tissue in angiosperms, and we don't really know the consequences of having this extra set of chromosomes. So instead of being haploid, it's triploid, but there are some hypotheses suggesting that might help with rapid development and also maybe some increase in genetic variation. In coconuts, for example, coconuts are angiosperms, and in particular, they belong to the monocots. That means that monocots are going to have one cotyledon, and here. So this is a monocot. It's in maize. They have one cotyledon or cotyledonary leaf in the embryo. That's the definition of a monocot. And the dicots are going to have two cotyledons in the embryo. So you can see this, this is one cotyledon and this is another cotyledon in the garden beans. And you also can see these two cotyledons in here in the castor bean. In this coconut, this is a fruit, but it's also a seed. And the seed, or the seed of the coconut, is going to have a triploid endosperm that is massive. All of the things that we eat from the coconut is triploids. That means that it's the endosperm. And the endosperm, it's at the beginning is liquid, and as it develops, it becomes harder. And it's this white tissue that we eat in cookies or in other products. Just to summarize, this is the life cycle of the angiosperm. So we have a plant that produces a flower, and in this flower, we're gonna have the pistil and the stamens. And the stamens are gonna produce the pollen, in particular in the anthers. The pollen will land in this pistil and the stigma. It will grow through the style, and it will reach the ovules. When that happens, we're gonna have a zygote. But keep looking at this ovule. Now the ovule is gonna transform into this seed. I say that ovules are unfertilized seeds. Once the fertilization occurs, right here, we have a zygote that is diploid, and you can see that it's dicot. It has two cotyledons. And the endosperm is going to be triploid. It's not marked in this figure, but in angiosperms, we always have a triploid endosperm. And then the integuments are going to form the seed coat. Now, please look at this fruit, because this looks a little bit like a peach. Hopefully you can think about a peach in this case. This is not the flower of a peach, but for a second, let's imagine that this is a peach. And the ovary, this is the ovary in the pistil. The ovary is the one that is going to transform in this fleshy tissue that we eat in the peach. And then the fruit is going to contain the seeds. Once that we eat this peach, then we can discard the seeds, and the seed can germinate. It will produce the embryo. It's going to have one end that is going to form this root, the root system. And these cotyledonary leaves are going to emerge in the stamen, in the stem. It's going to emerge. And then if the conditions are ideal for this plant, it will develop and it will flower and will close the cycle. So this is a recipe of my favorite salad, which has 1 cup of baby portobello mushrooms, 1 cup of arugula, 3 spoons of pine nuts, 1 spoon of coconut. You just have to mix and eat. It's delicious. You can add your favorite dressing, but based on the ploidy content, what type of cells are you eating? And please answer following the same order as in the recipe. Okay, so what's the portobello mushrooms? So portobello mushrooms are actually heterokaryotic haploid. That means that they are going to have 2 nuclei in these cells, but the nuclei are going to have a haploid set of chromosomes. So they're going to be haploid. The next one is arugula. Well, that will be diploid. And the coconut is going to be Oh, the 3 spoons of pine nuts. The pine nuts, part of the that we eat in the pine nut is the gametophyte, the mega gametophyte, and obviously the embryo that is inside, and the embryo is diploid. So it will be haploid and diploid, and then the coconut, as I just explained, will be triploid. So hopefully you get it right. This has a lot of information, so I expect you to remember all of this from the different groups that we have learned in class. Okay, let's go once more in this life cycle to emphasize the differences between angiosperms and gymnosperms. Okay, so this is the flower that we were looking at before, in which we have these 4 structures: the sepals, petals, the stamens, and the pistil. These structures, like the petals, we don't have them in gymnosperms. We also don't have closed or enclosed ovules, so we don't have this equivalent structure in gymnosperms. So in gymnosperms, we have naked ovules. In angiosperms, in the anthers, we're going to have the formation of these microsporocytes that undergo meiosis and form 4 microspores. And here you can only see 1. And then later on, mitosis will occur, and that will be the gametogenesis, the formation of the gametes. We're going to have a tube cell and a generative cell. The generative cell divides again through mitosis and form 2 spermatic cells. This is going to be important to remember because one of these spermatic cells fuses with the egg and the other one fuses with the polar nuclei. That's different than what happens in gymnosperms. In the ovary, this structure that is carrying the ovules, we're going to have an ovule that has a asperangium, which is diploid, undergoes meiosis and forms 4 megaspores. 3 die, 1 survives often. In most angiosperms, that's when it happens. And then through mitosis, remember this in blue is haploid. It's representing the haploid condition. But it doesn't mean that the whole ovule is haploid. It's just showing that something happened. That means the meiosis happened. And now we have this megaspore. And the megaspore is dividing through mitosis to form this mega gametophyte or female gametophyte that has the antipodal cells that are ephemeral, the central cells with the polar nuclei, synergids, and the egg. So 7 cells, 8 nuclei. And then, once that's ready, the pollen will land into this stigma. It will grow and carry these spermatic cells. The spermatic cells will reach the ovule, and the ovule will get fertilized, but twice, right? And that's one of the main differences between angiosperms and gymnosperms. Angiosperms have double fertilization. So the fertilization of the egg and the fertilization of the central cell. As a result, we're going to have a zygote that is diploid and an endosperm that is triploid. And the endosperm is the nutritive tissue that the embryo is going to use to germinate. So remember, this seed is this capsule that contains a very sacred and a special living organism that is going to contain enough food and the best protection ever. So then once that it reaches an area with the right conditions, it will germinate and it will produce flowers that later will produce the pollen and the ovules. So please take a couple of minutes comparing these 2 life cycles for you to notice the difference that I just explained. In flowering plants, after fertilization, the ovule becomes a blank, and the ovary, A blank. A, megaspore and megasporangium. B, pollen and spermatic cell. C, seed and fruit. D, stigma and pistil. E, fruit and dispersal structure. The right answer is C. In flowering plants, after fertilization, the ovule becomes a seed and the ovary a fruit. This is this is what happens. Ovules become seeds and ovaries become fruits. Flowering plants are an example of adaptive radiation. So during their evolution in the Cretacic, early on, they developed these strategies that I have explained, and that led to the diversification of this lineage. Currently, we have more than 300,000 species described of angiosperms alone. But then why are they so successful? And why they have diversified so much? We need to understand the flower development for that. And this is Arabidopsis thaliana. This is one of the model systems in plants. And it has these 4 whorls, the sepals, petals, stamen, and carpels. And this is an idea of how flowers developed based on the turn on or off of some regulatory elements, some genes that function as regulatory elements. And this is a simplified version that says that the activity of the A gene will be involved in the formation of sepals and petals. The activity of genes, the gene B, will be involved in the formation of sepals and stamens. The activity of gene C will be involved in the formation of stamens and carpels. This is, hopefully it's not confusing, but it will help you to understand that by turning on and off some of these particular genes, you can have flowers that lack sepals. For example, if you turn off this gene, you will end with flowers that do not have sepals. Here, I would like to add that this is a simplified model, but more advanced models include also the gene class E. So this is the normal flowering plant, and this was done based in some work using mutants and produced in the lab. And you can see that when you mutate the A type of gene, the activity of this gene A, then you end with a mutant that is lacking both petals and sepals. And you have only these carpels and stamens. Then what happens when you mutate the class C, the type of genes, the class C? Well, you have a flower that lacks sexual structures. So that has led this, playing with the turn on and off of this gene explains partially the diversification and the different and amazing diversity that we have in flowering plants, and not only in flowering plants, but also in gymnosperms. So the more than 300,000 species of angiosperms have wide, diverse floral morphology, mainly due to the different expression patterns of A, FOXP2 genes, B, BHLH transcription factors, C, genes of the ABCE transcription factors, D, genes involved in nectar production, or none of the above. Well, this is all for this class. Next class, we're gonna start covering diversity in angiosperms, and we're gonna learn more about the different groups that are in this very diverse group of plants. Thank you.